And so welcome back. We hope that you enjoyed the meal and you had fruitful discussion around the posters. So we are going to start on next session that Rodrigo, Almeida, and myself are going to co-chair on the vectors biology, actually. So our first speaker for this session is going to be Nicola Bodino from the CNR, Italy. And so you are a recipient of EFSA Young Researchers, and you are going to talk about reproductive biology and egg parasitoids of Philaenus spumarius. Thank you.
Thank you very much for the presentation. I'm Nicola Bodino, postdoc researcher of EPSP CNR in Torino. And I'm going to present some work about the reproductive biology of Philaenus spumarius. So given the experienced audience, I will skip altogether the introductory part about Xylella and vectors and jump to the topic of the presentation. So reproductive biology of Philaenus spumarius. Why bother to study it? Because reproductive biology of this insect is really quite understudied. Despite its potential importance for control of these vector populations. Very briefly, Philaenus spumarius is a monovoltine species, so one generation per year, and so it is characterized by a long adult life with two dormancy forms, an ovarial parapause during the adult stage, the summer period, and an egg diapause during the winter season. But the timing variation across these climatic and latitudinal gradients of the two dormancy forms are really not well understood yet. Also, very recently, Formisano et al., 2022, found some Italian population of Philaenus spumarius being associated with Volvakia, well-known bacterial symbiont, often known as a reproductive manipulator. Also, the association between Volvakia and Philaenus spumarius seems to be more frequent in colder climate, but this hypothesis needed to be tested. Finally, so far, only one egg parasitoid species, the Hymenoptera mimaride, Octonus vulgatus, has been found in the Mediterranean area of Europe. And so more investigation are needed to find out if there are more species or a more diverse community of egg parasitoids associated to this insect pest. So here I will present some preliminary studies and results on specifically the variation in timing of a reproductive phase of Philaenus spumarius, so to identify a molecular marker of a reproductive phase that can help to get precise prediction of the duration of the ovarian parapause during the adult stage, and the precise timing of the egg maturation and then oviposition in the different climatic areas of a species that is very is a huge areal in Europe. Second, we investigated the variation in prevalence of Volvakia in different populations along altitudinal gradients. And third, we start to investigate the presence and composition of egg parasitoid community associated to Philaenus spumarius in northwestern Italy. So, about the first set of experiment about reproductive phase, we sampled adult Philaenus spumarius females from several localities in different latitude and elevation across Italy and in different periods of the year. Each Philaenus spumarius female was either dissected to evaluate the developmental stage, so the number of developing or mature eggs inside the ovaries, or subjected to molecular analysis to measure the expression of the vitellogenin gene. So the results showed clearly that the number of developing eggs in the ovarioles of Philaenus spumarius females was well predicted statistically by the expression level of vitellogenin. That is, vitellogenin expression level is a good estimate of ovarian development. With this information in mind, we can observe that the vitellogenin expression peak is way earlier in Philaenus spumarius from localities at high altitude, 2,000 meters, 1,500 meters, compared to those in lowland or lower latitudes. So the vitellogenin expression timing follow an altitude-latitude inverse gradient. So the duration of the ovarian parapause tend to increase as elevation and or latitude decrease, and therefore the average temperature tend to increase. Keep in mind this information I will discuss a little bit later, talking about the second set of experiment, Volvakia. We investigated the Volvakia prevalence in Philaenus spumarius females, sampling individuals in high altitude localities in Western Alps. And we confirmed the hypothesis present in the study from Formisano et al. 2022, so that Philaenus spumarius population at higher altitude present higher prevalence of Volvakia association. 68% versus 35% in lowland at the same latitude, more or less. Why? This is probably because clearly at higher altitude it's colder and there is a shorter parapause. The association between Volvakia and Philaenus is probably influenced by the parapause duration. The longer the parapause, the lower the Volvakia prevalence. This is probably influenced by the long summer condition that might reduce the persistent and the vertical transmission efficiency of this bacterium in the insect host, leading to a complete disappearing of this association in South Italy and hot Mediterranean areas. Okay, about the last set of experiment on the egg parasitoid communities, we exposed sentinel egg masses of Philaenus spumarius in several localities in northwestern Italy, leaving them during the winter for two different exposition periods, early and late winter. Then we retrieved from the field the eggs and observed the emergence of adult parasitoids. We found out quite good number of parasitoids, 92 out of 900 eggs retrieved from the field, but belonging, all of them, to one single species that already mentioned, Hymenoptera mimaride Octonus vulgatus, with a parasitization rate very variable among different localities from 0 to 26%. That is not so high, we have to say, but it's, by the way, interesting. So there are other investigations that are ongoing, so the molecular identification of this parasitoid to confirm that is actually Octonus vulgatus, and some parasitization trials that we started last year, but we are planning to continue in the next year of in control condition of Philaenus eggs, but also of other alternative hosts of Octonus vulgatus that so far are not yet known. So, in conclusion, the knowledge of the reproductive biology of Philaenus spumarius can be important to help the design of new IPM strategies that could add up to the current implemented management strategies to suppress, to limit the population of these insect pests. And this could be done exploiting the precise prediction that we got during this study of the oviposition time window, so useful to target the exact time of oviposition in different climate areas and to disrupt the egg-laying behavior, for example, with the crop plants or other strategies. Also, the exploitation of Volvakia could be interesting if the cytoplasmatic incompatibility would be confirmed. And finally, the enhancement of natural control by native parasitoids through a conservation approach could be interesting, also if we would be able to understand the environmental determinants of the presence and the parasitization rate of this species of egg parasitoids. And finally, this information can be implemented in population dynamics models or epidemiological model of disease associated to Xylella fastidiosa in Europe. Thank you for your attention.
All right, thank you, Nicola. So the next speaker is Julia Lopez Mercadal from the University of the Balearic Islands in Spain. And
thank you very much. This was part of my PhD thesis. So basically, we already know from other colleagues and other locations that they did studies on modeling which factors can influence the abundance or the presence or the occupancy of Xylella fastidiosa vectors. For instance, we know different climatic factors, the presence or not of the forest, the type of crop in where they are living, the sunlight hours, or the presence or not of the cover vegetation in the crops. So in the Balearic Islands, Xylella fastidiosa was detected in 2016. And since that moment, we started 2 projects. And we did an extensive surveillance in 3 crops, in olive, almond, and vineyard. And we confirmed the presence of Philaenos spumarius and Neophilaenos campestris in all the archipelago, in Menorca, Mallorca, Ibiza, and Formentera. Nowadays, the bacteria is almost spread throughout the archipelago. So with all this data that you can see the methodology of the vector transmission test and the sampling in this paper, we try to analyze the data with different models to see which can be these ecological drivers that can modulate their presence and their abundances in the case of vector surveillance. So for this kind of data, we did a zero-inflated model with different variables, as you can see here, the type of crop, the species, the vector species we were talking about, the temperature, evapotranspiration, the month of the month, the year, the week, the day sampled, the locality, the plot, because there were around 30 plots, the season, and the compartment vegetation in where the vector was sampled, meaning cover vegetation, canopy, or the surrounding shrubs. And the dependent variable was the spittle bug density. By the other hand, with the transmission assays, we tried to see if we had any factor that could be influencing the transmission. And we did mixed models with binomial type ones, with fixed factors with gender and species, and the random as a year, because we did different trials in different years. And the dependent variable was 1 or 0, and it was meaning the status of Xylella fastidiosa infection of those vectors. So first, I want to show you a bit how was the seasonality of the vectors in the islands. In the cover vegetation, we saw that there are 2 clearly peaks of insect abundance, one in a spring related to the vector the vector starting the cycle in this year, and then the second part in the last part of the year when they are mating in the cover vegetation. Regarding the surrounding shrubs, we know that they are mostly in summer, in the summer month when the cover vegetation is drying. And then again, same pattern in the canopy of the trees with two peaks. So what the model showed us is that for Philaenus spumarius, the presence of the vector was positively influenced with the, if there was tree canopy and cover vegetation, as well most present if the crop was vineyard, and in a spring, and very favored for the raining. By the other hand, it was negatively favored for the summer month when we know it's very scarce. In the other hand, for the abundance of Philaenus spumarius, we saw that when the vector is present, it's more abundant in almond crop than in the other crops, and that temperature and evapotranspiration has a negative effect on their abundance. In the case of Neophilaenus campestris, the models show that it's more present in autumn and in the cover vegetation, and that is not favored for the temperature. And regarding the abundance, we see that it's more abundant in the surrounding shrubs and in the almond trees, as well for the autumn season, and again, negatively affected for the temperature. Regarding the transmission test, here is, I wanted to show you some of the results we had. So basically, around of the 20% of the insects collected from the field, they were positive. And we could see the inoculation of the bacteria in the 15, 30, 45, and 60 days after the inoculation period. And when we modeled this data, what we saw is that the prevalence of Xylella fastidiosa in the vector was not depending on the vector species, so if it was Philaenus or Neophilaenus, neither on the gender. And that the prevalence of Xylella fastidiosa in Medicago sativa after the inoculation does not depend, again, with the vector species used, neither on how many of them we were putting in the plant to infect. So finally, the take-home messages or conclusions is that temperature and evapotranspiration affect negatively to the vector abundance, that precipitation seems to favor their presence, that cover and tree canopy favor the presence of the vectors, that there is an important preference on the type of crop for them, and that both Philaenus spumarius and Neophilaenus campestris have the same transmission capacity. Finally, I would like to acknowledge EFSA that we could do this thanks to a grant, and to all my colleagues that supported me during all the samplings and the trials. And thank you for your attention.
All right, thank you. So the next speaker is João Lapis from University of São Paulo in Brazil. And as João is walking here, I will tell you that João is the person who introduced me to science, and there is something really cool about introducing the person who introduced you to academic research. And ironically, I ended up working with him because the plant pathology department said I was not qualified to work for free. So with that.
Thank you for introduction, Rodrigo. Okay, this afternoon I will talk about the work of my former student, Joyce, and also which is continuing into her postdoc research. We are talking when we talk about vectors in Brazil, usually we speak about sharpshooters, but today my focus will be a little on a spittlebug. So, as you know, olive in Brazil is a very young crop. In São Paulo is growing in higher elevations because of where we get lower temperatures. They are facing problems with Xylella. The disease is very bad there. There's a lot of vectors, many species. So the questions we are addressing is on ecology of this vector is to try to understand aspects that allow us to identify those vectors that are more relevant. And some of the parameters we are testing is the prevalence in the crop, occurrence of reproduction, population densities, natural infectivity, and transmission. We have done transmission experiments for several of species, and for others we are still studying, such as the one we are going to be focusing today, the Clastoptera, which is a spittle bug of the Clastopteridae family. So in previous work, we did an extensive survey over several years with different methods, yellow stick traps, and also sweet nets on the ground cover and on olive canopies, and in different, a range of altitudes in the region. And we found almost nearly 100 species, but 18 were characterized as the most important in terms of faunistic indices. And these three of them, two sharpshooters and one spittlebug, Clastoptera, are important because they are prevalent in all the locations we sampled. Clastoptera is a spittlebug. As you see, it has a high density, average over these 5 years. It's the third largest. Compared to the other two abundant species, it has a population peak that is more skewed to the right. Has a population peak more on the fall, late summer, in the different locations. A more interesting aspect is about the occurrence on the canopy of the trees. So when you sample the branches of the tree, it appears as the most frequent species as adults. But more importantly, it colonizes. So you can commonly find the spiros, indicating the nymphal development, and also adults, as you see in the picture to the right. So it's the only species that we commonly find in olives, and I think probably the only one that consistently colonizes olives. When you analyze these insects for the presence of Xylella, a lot of them have Xylella, and Clastoptera also has it. So indicating that it's a natural carrier of the bacteria. We haven't done experiments for transmission. We are carrying out them. We don't have the results right now. The ground cover, we also have some species that we need to understand. But talking about the host plants of these spittlebugs, now we know very little about the biology. Actually, the taxonomy of these bugs is still needs studies. So Joyce will work with Vinton Thompson, which is a specialist, to characterize, because we thought we had like 4 or 5 species, but apparently it's only one, but it's a new species. So we need to identify this Clastoptera, which we call now SP1. But besides olives, it has other shrubs and trees that are harboring this species. We find nymphs in Duranta, in Salvia, in Rhododendron, and other plants around the orchard. These are a number of spiros, so it's data that she collected recently, indicating that the Duranta erecta and others are developing hosts of this plant. So, and probably they are hosts of Xylella too. That's the next step she's planning to do. So in conclusion, we can say that Clastoptera SP1, this new species, likely plays a role in Xylella fastidiosa spread because it's a prevalent species in orchards. of this main growing region in São Paulo and Minas Gerais. Adults and nymphs colonize olives on a number of shrubs and trees from different families too. So it apparently has a wide host plant range around the orchards. And also adults carry Xylella fastidiosa. So what we need to do now is first confirm the identification of this as a new species and have the name, and also assess the ability of transmission of Xylella to olives. Thank you. This is the group of collaborators that are helping this project, students, and thank you, the financial supporters.
All right, thank you, João. So our next speaker is Lindsay Burbank from the USDA ARS Parlier Station, which is the main group, USDA group that studies Xylella in the US.
Thank you. So first, I just want to point out that this is a team project that I did with my colleagues also from the USDA, Dr. Rodrigo Krugner and Dr. Elizabeth Rogers. So I'm here from California in the United States, and we have a lot of Xylella in California. We have for a long time, and mostly what we find is subspecies fastidiosa, and then the subspecies multiplex. And so the main crop diseases that we deal with in relation to Xylella are Pierce's disease and almond leaf scorch disease. What we don't have in California is the subspecies pauca causing severe disease in olive. And so this is not something we have in North America. It's never been found there. But it's very much something that people are thinking about and people are concerned about. In particular, the olive industry in California, which is much smaller than the industry in other countries, but we do have an olive industry in California. And they're concerned about what would happen if this disease were to show up in our area. And part of the concern is that we know we have a lot of vectors of Xylella in California. These are two that have been associated with mostly Pierce's disease, the blue-green sharpshooter and the glossy-winged sharpshooter. But we have more species of insects as well that can transmit Xylella, and we've seen what they can do as far as spreading it around in our agriculture. So the goal of this project was to get some information specific to the olive industry in California to help them understand what the risk would be if the subspecies pauca ever does show up in our state. And so first we just did some evaluation of some of the important olive cultivars in California to see how susceptible they would be. And then also we were testing glossy-winged sharpshooter to see what the acquisition efficiency and potential for transmission would be in relation to subspecies pauca in olives. So first, a little bit of information on the California olive industry. Again, it's much smaller than what other countries have, but it's been around for quite a while. This graph on the left shows olive production in California since the 1980s. And the green part of the graph is olives that are produced for table olives. Mostly these are canned ripe olives that we produce in America. And then the red bars show what's produced for oil production. And so historically, you can see most of the olives produced in California were for table olives. And then more recently, we've started to get an increase in the olive oil production. But table olives are still a big part of the industry in California. So what's important about the cultivars is that the table olive industry specifically in this area, they really just use a very small number of cultivars. And so the pie chart on the right shows the predicted olive harvest from 2022 in relation to the cultivars that they're growing. And you can see that over 90% of it comes from one single cultivar, which is a cultivar called Manzanillo. 7% comes one called Sevillano, And then less than 1% comes from everything else that they're growing. And again, this is specific to table olives. And these cultivars have been in California for a long time. They were brought in around before 1905, most likely from Spain, and they've been grown there ever since. So we tested these cultivars to see if they were susceptible to the subspecies Pauca, ST53 from Italy. And we tested Manzanillo, Sevillano, and then the Mission cultivar, which is produced for both table olives and for oil. And all three of these cultivars are in fact susceptible, so this is bad news for the industry if they ever get this disease. You can see that in the photo there's some dieback that shows up in the plants. This is after 1 year post-inoculation. And so all three of these cultivars were susceptible. So the second part of this project was to look at the glossy wing sharpshooter and what its potential as a vector of subspecies Pauca in olive would be in relation to the California industry. So a little bit of information about the glossy wing sharpshooter. The map on the left shows the current range of the glossy wing sharpshooter. This is an invasive insect in California. And the blue areas are the areas where it's already established. And then the green areas are places where there's a risk for it to be established. And then the map on the right shows the major olive-producing counties in California, with the circle around Tulare County, which is the #1 olive-producing county in the state. And you can see that this overlaps with the current range of glossy wing sharpshooter. So if we're thinking about the vectors that would be important, this is definitely one of them. So we did some acquisition experiments using seedlings inoculated with subspecies Pauca, ST53. We caged glossy wing sharpshooters on these plants for a 3-day acquisition period. And we did this with the 3 cultivars, Sevillano, Manzanillo, and Mission, with about 15 plants per cultivar and 2 insects on each plant. And then we repeated the experiments at several different time points, so after 30, 60, 90, and 100 days post-inoculation of the plants. Then we did the entire thing a second time with new plants. So in total, this is about 700 to 800 insects that we tested in the acquisition experiments. And we tested all of the insects with PCR directly after the 3-day acquisition period. So what we found is that, not really that surprisingly, glossy wing sharpshooter can acquire Xylella from infected olive plants of these 3 cultivars. So this is broken down by cultivar. The acquisition rate was relatively low in the experiments. It was somewhere between 2% and at the highest 10 to 15% of insects acquired in the 3-day period. And then if we look across the entire all of our 700 to 800 insects, it was about 4% overall acquired Xylella after 3 days. And so this is just the same information separated by time point. So we did the acquisition experiments at different times after inoculation of the plants. And we did get some acquisition as soon as 30 days after inoculation. These are seedlings, so they're very small plants. And then we did see acquisition throughout the entire time period. The last time point was at 180 days or 6 months after inoculation. So this is some important information for the olive industry in California. It's also hopefully interesting to others who are looking at either potential introduction of subspecies Pauca or potential introduction of the glossy wing sharpshooter. And we found that the California ripe olive cultivars are susceptible to the ST53 and that the glossy wing sharpshooter can acquire Xylella after a 3-day acquisition period. So with that, I would like to thank the funding for this project. This came from the California Olive Committee, California Department of Agriculture, and the USDA, the U.S. Department of Agriculture. Thank you.
All right, thank you, Lindsay. The next speaker is Alberto Ferreira from CSIC in Madrid. And here's
Thank you very much, Rodrigo. I will be talking about degree day-based model to predict the egg hatching of Philenus. So, as you already heard today, Xylella fastidiosa is a bacteria that causes severe disease epidemics in the Mediterranean, mainly in Italy, also in Spain, Portugal, and Israel recently, and Lebanon. In Spain, it causes problems mainly on almonds. So these are the symptoms that you've been looking at in the previous talks. Xylella is restricted to the xylem, but in the vector, it's retained in the Presivarium. And the main vector in Europe is Philenus espumarius. So there are other species that can transmit the disease, spittlebugs and sharpshooters, but Philenus espumarium is the main vector in olives. So just going a little bit into the life cycle of the Philenus espumarius. It's a univoltine species. It lays the eggs in October to February, quite a long stay period of time. The nymphal stage can develop between February and May, depending on the regions. This is related to the cycle in Spain, I would say. And the adult emergence starts in the beginning of March, and you can have adults until April. Then we have the adult stage, which will live in the natural vegetation, herbaceous vegetation below the olive trees. By the way, in Spain, it's difficult to find Philenus espumarius in the olive canopy, but it's abundant in the grasses, in the weeds below the trees, the olive trees. And then during the summertime, it disappears completely and it goes to oversummering woody hosts such as oaks, pines, and shrubs, where we can find it. This migration occurs during June, July, depending on the areas. And at the end of the summer, in the beginning of October, we will get migration to the emerging vegetation after the first rains in the olive orchards. So this is the life cycle. This is when the transmission occurs, mainly during the summer migration, where you get the secondary spread of the disease when they visit the plants from tree to tree. Even with low populations, you can have spread of the disease. But one of the questions we don't know about the life cycle is when do the eggs can hatch, and why do they hatch, and in which specific time of the year they may hatch. So based on this question, we wanted to understand what was the temperatures needed for the egg to evolve from the egg stage to the newborn nymph. And for that, we developed a degree day-based model to calculate the degree days accumulations to get the first nymphs and predict when would be these nymphs be emerging in different areas. So for that, we developed this degree day model, which was based on systematic samplings in different regions of Spain and also on field observations that we did in different places around Madrid. So the final goal of our project was to build a decision support tool that could predict the best timing for Philenus espumarius management. As you know, the most vulnerable stage of Philenus is the nymphal stage. So that was the target of our work. So for that, we did sample basically egg masses that were produced in October to November in different dates, and we placed different cages at different altitudes where we monitored the evolution of these eggs based on different sites at different altitudes around Madrid. And then we, just before hatching, at the beginning of spring, we moved these eggs to plants where they were with Sonchus plants, so the newborn nymphs would be able to survive. And we monitored the egg hatching date of these different egg masses in those different sites. So then we constructed a model based on degree days, And we tested also different diapause ending, because as you already heard, this insect has a neck diapause, but this may be different from one region to another. So we tested different diapause ending dates. Finally, we found that December 1st was the best fit for our data. And then we calibrated the model using different temperatures, maximum, optimum, and base temperature, minimum, maximum temperatures, to have the best experimental data fit. And then we validated the model using field observations in different regions of Spain, in Spain, where we had monitored these nymphs during different years. Then the model was able to calculate the precise timing for controlling Philenos spumarius nymphs most efficiently. So that was the final goal of the model. By looking at the results of this study, you can see that 435 nymphs were hatching in different periods of the time of the year, and in different sites there were different hatching dates. But most of the hatching occurred during early March to early May. If you look at this map, this is a simulation where you can see the predictions of the model based on, and the white dots represent the real field observations that we had on year 2018. And in the model, you will see what would be the predictions of hatching probabilities at different parts of Spain, starting from the south and moving to the north. So with this model, and by setting up the diapause ending date in December 1st, we could predict very well the field data. Based on that model, we developed a decision support tool that will help farmers, growers, on how and when to treat, on when to act against the Philenos spumarius nymphs. So based on this model, and based on that hatching days may occur in 2.5 months, nymphal development may happen during 5-6 weeks, the model predicts that 2 different action dates would be the best to control most of the nymphal population of Philenos, and that in most cases, with these 2 treatments, you will control most of the population. Of course, you will have to control this when most of the eggs have hatched, but before the nymphs reach the adult stage, to have a real efficacy of the treatments or the control actions. So in this case, for example, for the north altitude, with the first treatment, you can control 40% of the population, and with the second treatment, you will control 90% of the population. So this tool has been developed. It's available. It's publicly available, and depending on you can access this site, and you can just introduce the data, meteorological data, or the observatory, and you can have an idea on when should be the best moment for controlling Philenos spumarius eggs, depending on temperature. So basically, the take-home messages, I would say, is that the temperature is a critical factor that impacts the hatching of Philenos spumarius. It's essential to understand the diapause of Philenos spumarius, which may change from one region to another, and depending on different variables. There will be, considering the December 1st as the ending of the diapause, the model is consistent, and it predicts well the observations of egg hatching that we have in Spain. And applying control measures on 2 different days allow to target the highest number of nymphs. Then I would say that our decision support system can predict when would be the best timing for acting against Philenos spumarius, depending on field site. You can be able to introduce your field site, and it will predict you the best timing of control action. And this, if you would need more information, you can follow up in a paper that was recently published. And I would end up my talk acknowledging the Institute of Physics in the Balearic Islands, and also, of course, our group in IFB, Institute for Agricultural Sciences, and our team, and of the granting agencies that you can see below. Thank you very much.
All right, thank you, Alberto. Okay, and our last talk for this session is by my co-chair, Astrid Cruel, from INRA in France.
Thank you, Rodrigo. So actually my talk is kind of a link between this session and the next session about surveillance, because I'm going to talk about vectors as sentinels of plant diseases, and I'm going to talk a little bit about the impact of climate change on prevalence of Xylella in vectors. So as you know, monitoring of Xylella is usually carried out by analyzing plants, and especially symptomatic plants. But in the wild, plants are frequently asymptomatic, and then there is a number of drawbacks for targeting plants only, as the bacterium is heterogeneously distributed within tissue and some other inhibitors, etc., etc. So the idea would be to generalize the use of insect vectors as sentinels to complement plant-only surveillance. And as you know, so Philenus fumarius is kind of the perfect sentinels, as it is largely distributed in Europe. So back to 2016, we did a first test in a few populations across Corsica in France. We used nested PCR and Sanger sequences, and we showed that Xylella was widely distributed across the island, even in areas that were not recorded by plant surveillance. So we wanted to go one step further because not much is known about how the ongoing climate change is going to affect the epidemiology of such generalist vector-borne diseases as Xylella. And the question here is especially relevant as this increasing temperature may impact both the biology of the vectors and also the biology of Xylella. So we monitor the prevalence of Xylella within population of Philenus fumarius in climatically contrasted areas in Corsica, and we try to answer, okay, do we observe any influence of some climatic factors on this prevalence? So we did a 4-year survey, 39 population of Philenus fumarius, about 1,200 specimens that were screened for the presence of Xylella. So we improved a bit the methods as compared to what we used in 2016. We use a 2-step PCR approach for replicates for each specimen, and followed by MySeq sequencing. So basically you do first PCR in which you target the region with specific primers flanked by tails. And these tails enable a second PCR reaction to add Illumina adapters and unique combination of indexes to multiplex your samples and reduce index hopping. Then you sequence your data and you analyze with a bioinformatic pipeline. All the protocol pipeline is available in the publication. So for the climatic profile of the sampling sites we have followed, we use 25 temperature-related and 5 precipitation-related variables, so altogether 30 climate-related variables. So we split each year into the growing season, March, November, and the dormant season, December, February. And then, according to the phenology of Philenus fumarius in Corsica, we first split the growing season into March, June for the larvae and the young adult, July, August for the estivation period, and September, October for the adults. For the temperature variable, we use point estimates, but also average, minimum, maximum over the time slice to better describe like prolonged thermic stress. So we use daily mean temperature, max, and average daily max. We also look at the number of days with temperature superior to 16, 18, 20, etc. degrees Celsius. And for the dormant season, we use the number of days with temperature inferior to 0 to 4 and 6 degrees. And for the precipitation, we use the sum of daily precipitation, and also we don't have a lot of data regarding the impact of precipitation on Xylella fastidiosa, but we have one study on Pierce disease that showed that the incidence of the disease is higher following high rainfall years. So we use the sum of precipitation in growing season of the year y-1. So we analyzed this data with statistical methods, PCA first, and also a partial least square regression analysis. So on the left part of the screen, you have the results. So 1 column for each year, actually. So we didn't follow the same population across the 4 years because we wanted to reduce the interdependence between the different data. So the black portion of the donut is the proportion of Philenus plumaris from which the bacterium was sequenced. And the color is the score of the sampling sites on the first PC axis of the PCA conducted on the climate variable. So in fact, the warmest the color is, the highest the temperature was. So the results between the two models are quite similar. They show that the prevalence is positively correlated to temperature. And in the second analysis, so the PLSR analysis, you have one more data that shows that the prevalence is actually predicted to be higher under milder winter. But none of the precipitation-related variables were retained in our models. So how can we explain these results? So based on the literature, we can say that very likely these milder winters, they increase the overwinter survival of Xylella, and the bacterial load consequently is higher in spring. The multiplication of Xylella is favored during the growing season because you have higher temperatures, so you have high cell density and biofilm formation is achieved earlier, so the acquisition by insects happens sooner. Then, because of high temperature, the vectors, they may fly more frequently, they may disperse further, they take longer meals, so the probability of encounters between vectors and Xylella is increasing, and the acquisition and transmission of Xylella fastidiosa is favored. So we have a lot of open questions. What is the overwinter survival of Philenus plumaris, for example? Has temperature have an impact on the increase of population sites, etc., etc.? So a lot of questions to answer. So what next? We also use some scenario of global change. So under 3 shared socioeconomic pathways and 5 global circulation models, the first thing we can see is that, so here you have the past temperature of the coldest quarter of the year, and here the model, so you can see that with time, every model predicts that you will have an increase in this winter temperature. And in parallel, the species distribution modeling show that both Xylella and Philenus will keep encountering suitable climate condition in the island. So the yellow is the most suitable, the red is the less suitable. But this suitable condition could be, so the condition could be less suitable on the coastal area and more suitable in higher elevation areas. So you could have a migration toward higher elevation area. Could we experience a system runaway? It's hard to answer this question, but my feeling is probably no, because first, we have an argument against that, which is the biology of Xylella fastidiosa itself. It's more commensal that exhibits self-limiting behavior through quorum sensing. And then we also have some data, but actually few, that shows that vectors, especially in the US, they prefer to feed on healthy or asymptomatic plants in controlled conditions. But what we ignore so far for everything is the adaptation to climate change for both partners. And we cannot take this adaptation into account into all our model. And what we should avoid as much as possible is, of course, the possible introduction of more efficient vectors to European Union, and then also new strains. So just to conclude, so the study confirms the power of insect vectors to track Xylella in ecosystems, I think, and my feeling is that just to go beyond Xylella fastidiosa, I'm pretty sure that with these new tools we have, and especially the progress we made during the COVID epidemic, with the PCR machine, with the man-ion sequencing, we could really set up and develop new tools to complement plant-only survey for Xylella fastidiosa, but also for other vector-borne plant diseases to better understand, just for surveillance, but then also to better understand the driver of this plant spread, how they had established, and what they and how they will evolve. So if you are interested in discussing that, please don't hesitate to contact me. Thank you for your attention. So we now have time for questions.
Please raise your hand, stand up, and say who you are, because we really cannot see you from here.
I have a question for Julia and Alberto. Given that seems to be quite, the local conditions quite a very important factor for Philenus distribution and hatching and whatnot, and lots of different conditions affects, different factors might be affecting different ways. I guess the question is, did you have a chance to validate the models in the field? But the main thing is, if there's something in common that drives the population size of Philenus, I understand that obviously the temperature needs to be, it's a big factor because first they have to hatch, but then when they establish and how persistent they are and what sort of triggers them to move potentially into the crop and whatnot. So if it's something that is common for Philenus or maybe for spittlebacks across.
Yes. Do you hear? See. So, when I was doing the models, performing them and selecting them, once I selected the one that was supposedly statistically correct, right? I tried to see if it was logical with what I was seeing, because before doing the model, I spent more than 3 years in the field going to sample them every week. So I could really see that it was a confirmation of what I was seeing in the model as well. So basically I would say that the most important is the temperature and as well precipitation, because when temperatures were very low or were very high, you could not find the insects in the field. As well, of course, when it was raining, it was one of the factors that supposedly should favor the insects, because I guess it's related a bit with the temperature, because when it was, for instance, raining in summer, temperatures were a bit mild in comparison of the rest of the summer, which in Balearic Islands is very, very warm. So first, this is very important. And then for me, of what I've seen, maybe some colleague wants to add more information, is the landscape composition. This is very, very important. So if, let's say, how the plot that are you sampling is composed, like how is the management of the cover vegetation, how is the management of the crop you are studying, if in the surrounding area you have a forest or you have nothing, or maybe you have oaks. This really is modulating the insects that you can find in the place that you are sampling. I hope I answered.
Domenico Bosco from the University of Torino. I have a question for Astrid and one for Julia. For Astrid, I fully agree with you with the fact it's very interesting the use of insects as a sentinel of the presence of Xylella in the environment. But this point was already debated for because of a problem. The insect can be, Xylella can be detected in an insect, but if you are doing a monitoring program in an area which is Xylella free, then the problem with the insect is that you cannot be back to resampling to confirm the actual presence of the bacterium. And I think that if you have to take decision also for regulatory reasons and so on, the detection of Xylella in an insect is not as reliable as in a plant that you can label the plant, you can go back, you can resample and confirm this. What do you think about this?
But you can go in the same place and try to sample other insects to confirm your results or not, right? And then you can also fail to sequence Xylella from a plant because you collect a branch in which the bacterium is not present. So I'm not kind of saying that we should replace plant surveillance by insects. I'm trying to say that we should complement plant surveillance with vector surveillance. And I would go for, to me, vectors enables kind of a blind surveillance. You have no a priori on the symptoms on the plants. My experience, it was a long time ago, but my experience was that the agency in charge, they preferentially targeted symptomatic plants, so there was a bias towards symptomatic plants. And so we miss, because this is what we show, we miss some places in which the bacterium was. And if we could have had in the past maybe kind of a global surveillance across Europe using vectors, everywhere in Europe, using vectors to try to have like the point zero of where we are concerning the epidemic. I would have done something like that. So I'm not advocating replace, just to complement.
No, I understand. Thank you. And for Giulia, you said that the presence of vineyards in the landscape, I guess, if I understood well, is a factor that increased the likelihood of having high population densities. Did you consider in this model the presence of alternative cropping plants other than vineyards? Because, for example, in Italy, in central Italy, colleagues from the University of Padua, Verona, they demonstrated that at the landscape level, the population size of Philenus spumarius correlates more with the presence of olive rather than vineyards, so that vineyards shouldn't be so good as a habitat for the insect.
Yes, I know, and of course I noticed that. But in this case, what the model was indeed saying is that it was not about that it would be more abundant. It's just that when with the 3 crops I selected, I sampled, when present in the 3 crops, it would be supposedly more present in the vineyard. But we have to take into account that the variable in the model, let's say, it was including as well like cover, canopy, and shrubs. So I think that it's as well related that when I was sampling in the vineyard plots, let's say in the canopy of the vineyard, the vector was very less abundant, but was very, very abundant in the surroundings. So I think it's because of that. It's not because it's more abundant because it's vineyard. It's just in these plots, the abundance was more present in other compartments of vegetation. But I don't know, maybe there are more things that could be affecting in there. But yeah, we should explore, I think, this data because I think it's quite interesting.
If I may intervene, I think it's kind of a mess now, the landscape information that we have, how it can influence the presence, abundance of Philenus and Neophilenus, because there are different studies that say quite different things, Maybe because from different part of Mediterranean and not only part, but it's difficult to find really I mean, probably in lower latitude, hot summer Mediterranean region, something that is not drying up is for sure attractive, whatever. The same does not apply in Central Europe or North Italy even. We compare the two. But I mean, maybe this is a general rule, but with many little variation that we are still far from really grasping. I really like the work from Sharpshooter 2022. That was really, I wasn't convinced about the of cistus in the quite strict association with Philenus association with cistus, but that paper really make me change idea. So very good.
Perhaps I could add that we might be dealing with different variants or haplotypes of Philenus, because when you look at the literature, you see that Philenus, it's in southern Italy, it colonizes the trees, the canopies, while in Greece, for example, or Spain, it's very, very difficult to find a Philenus on olive canopies. So this means probably that we have different haplotypes or variants of Philenus around Europe, which is making this controversy. This is why you find a lot of Philenus in one crop, and in a different crop you don't find in a specific region, and in the other the things change. So we might have more than one kind of Philenus, I would say.
But if I may add, we are indeed dealing with very different landscapes and situation in all of these places. I mean, I didn't sample in the peninsula or in Italy or in Greece, but I think that the point of starting is really, really different. I mean, for instance, I found a lot of insects in olive canopy. So, I don't know. Or maybe it was the manage of those plots. Who knows?
Continental Spain, as far as we know, not only myself, but many other people have been looking to Philenus in canopies, in olive canopies, and they can't find them. So it's difficult. It's different, yeah.
Mike?
Mike Jager from Imperial College London. You mentioned the preference of the vector for asymptomatic plants rather than infected plants, diseased plants. I just wondered how strong that was, because surely, given the presence of Philenus carrying bacterium, if there's a preference for asymptomatic or healthy plants, that will increase the potential inoculation rate and would lead to potentially a runaway situation.
That's your data, right? No? This is not. This is a study by Krugner. No, you didn't. No, I mean, let's say you were not on the, okay. So you were, sorry.
That's my data. Well, Matt Doherty's data, and analysis, and writing. Anyway, my name. But this has also been shown by João in Brazil. So there is this discrimination against symptomatic plants, which we've done this in California in different ways, even painting leaves and all of that, just to show it's a color kind of component to it. And we think that is linked to water stress, so the insects can kind of tell that apart. What we have been able to show that work that Matt led was that so essentially symptoms go up after bacterial populations in the plant go up. There's a lag period there. So there's a sweet spot for acquisition just when symptoms start to appear, when we have enough bacterial populations in the plant, and just before they start to discriminate. After that discrimination, they start to discriminate. I mean, it's not 100%, but they do start to discriminate. And bacterial populations continue to go up, but they don't like those plants anymore. So that's why you would see this kind of intermediate stage in a season, let's say, where you would see more spread, but then when symptoms show up, theoretically, you would see less spread. Having said that, these were controlled experiments, but the insects had choice. So we had uninfected and infected plants. They could choose whatever they want rather than just a single plant, no choice, which we know no choice experiments are great, but you can't infer anything from them related to behavior and how that would impact things. Does that sort of answer your question, or I forgot the question?
The main point is, even if there is only a small amount of, a small number of the vector carrying the bacterium, if it then favors landing, feeding, probing on asymptomatic or healthy plants, then that will likely increase the overall inoculation rate. So the fact that it's favoring the healthy plants, I mean, if it's carrying the bacterium and it does that on a diseased plant, effectively that's a dead end. But if it's favoring inoculation of the healthy plant, then that potentially, depending on that balance between what it favors, it could lead to a runaway situation. So I think, we can make too many assumptions on some of the results of these preference tests.
Yeah, I agree. I do not remember the specifics of that work, but I will say that some of it is done in the context of grapevines in California, and late season infections will not survive the winter. So there is that as well as a kink there. For that in particular, that Matt may have incorporated into the study or not, I do not recall. Mark Sisterson at USDA has done some work that's somewhat similar. You're probably familiar with that. I don't remember the details. Lindsay might. Okay. Lindsay's not wanting to talk about insects. So, yeah, so, I mean, you're correct. But the stuff that Matt has done suggests that it would not be some sort of runaway situation, but I do not remember why.
This is Saskia Okenhout from John Center. I would like to follow up on what Alberto Ferreras just said, is that we do find genetic differences in Phalaenus spiromeris populations across Europe. We have genome sequence data for 300 and more specimens from all over Europe, and there's a clear difference in genetics, and especially between southern Spain, southern Portugal, and the rest of Europe. So, and with that, I also would like to ask a question to Nicolo. And you found Volvakia in certain, I think, Phalaenus spiromeris at higher elevation. Is that right? You find Volvakia in Phalaenus spiromeris at higher elevations? Yes. Yeah. So I would like to follow. So Volvakia is maternally inherited, isn't it?
Yeah.
It is Volvakia.
Yes.
So I would like to say that also the Phalaenus spiromeris in higher elevations and in colder climates in general belong to the same mitochondrial haplogroup. And so it may also be that Volvakia is associated with maternal inheritance rather than being adjusted to cold climate. You know what I mean? Because it's maternally inherited.
Yeah.
Via, and so maybe via the mitochondria also maternally inherited. So maybe those lineages that have Volvakia and the haplogroups that are in higher elevations and colder climates seem to be more ancient. You see, so I don't know. So Volvakia might be an early infection.
I don't understand really well.
Okay, all right. Okay, well, I can.
I'm sorry.
Yeah, all right.
Saskia, we are now starting together with a colleague the genotyping of this population.
Yeah.
Together with Massimo Giorgini in Napoli, and so we can infer something also about the, let's say, genetic affiliation of this population that are more associated with Volvakia compared to the others. So I think we will be able to provide some more information in short.
Okay. All right.
So not only associated with the environment, but also eventually to haplotypes.
We have this information, I'm just saying. We have some information that may be also helpful. We can talk, yeah.
Thank you. Maria Saponari, CNR Italy. I have a question to Lindsay, sorry if it's on the vectors. Do you have some comparative data on olive infected with multiplex, and which are the rates of acquisition and transmission with Glossy wing Sharpshooter?
Yeah, so the multiplex was done by Rodrigo Krugner previously. The rates were pretty similar. They're also pretty low with the multiplex. But multiplex doesn't cause a lot of disease in olive, so we weren't sure if that was just because there was not a lot of bacteria in the plant, but it seems to be about the same.
And sorry, which is your feeling about the rate of acquisition and transmission you got compared to what you have in other crops, in grapes in general?
I don't know anything. We couldn't look at transmission with the Pauca because of the conditions of our quarantine permits. But I don't know what the comparison would be to grape.
Questions? We have lots of time. All right, since you don't have a question, I'm going to steer the plot here. So we had at least 3 of you talking about philenus and phenology and related things. And let's forget about everything you disagree on and other people here. And the really key for management is, when do you see the little guys out and coming out there at that point and getting rid of them, right? So we know at different times of the year when you're going to see them, some temperature models and whatnot. So are we at the stage where for the Mediterranean area, you can come up with some sort of very good guidelines where people can be like, temperature is up at, here the accumulation of degree days you have. This is when you should start seeing second, third instars, which means you still have a couple more weeks before you have to do anything. Maybe you should go out there and check and do something before you have adults. Are we at the stage where that can be done, which is a very, in some ways, simple, theoretically simple kind of recommendation? Or are we 10 years after the beginning of this, do we still need to do a lot more work to answer what in many ways is a very key question, and in many ways is a very simple question, again, theoretically, not in practice, but where are we in having that recommendation for people on the ground where they can actually take action?
Okay. I think that we are quite close, because what we've been trying to do in the last 2-3 years was to try to find out when should you treat, because you should treat the nymphs some way, or you should act in a way to reduce the population. And the question is, when are the eggs hatching? Of course, this depends on temperature mainly, but might be other variables also involved. But the point is that by using temperature alone, you could predict when would be the hatching in a specific place or site. Of course, you need accurate temperature, because there might be microclimatic conditions that may change, and by using just general stations might not be good enough. But if you have an accurate temperature monitoring system that you will be able to measure the degree days that are accumulated in a given region, you will know when to treat at the first instance. Of course, we found out with our model that by treating once is not enough, so you will have to treat twice. perhaps at the beginning when you have the first nymphs, but then when you have most of the nymphs in the field, and you have to treat in a way that you will do these two treatments in the specific timing, and this model can predict when should be this done, depending on the temperature in a specific site. So yes, by using modeling, I think we can quite target a high number of nymphs to reduce the populations effectively. Yeah, we are quite close.
All right, Julie?
If I may ask, well, I think that I just can speak about the Balearic Island situation, let's say.
Oh, no, this is an Europe question.
Okay, so this is such an exam.
It's not an exam, it's an Europe level question though.
Yeah. Okay, so I think that indeed we are really close. At least we have the general guidelines of how we have to manage the field, at least. We know that we could do weed management in spring and in autumn, and that this could help in suppressing the populations. And as well, you can apply insecticides, and we can have a lot of temperature models that can give us some hints of what we can do. But in my point of view, what we still need to know is that how can we do this to be really effective? So I think that the problem is to really know the effectivity of what we are doing, because at least the populations are really not very, very high to, for instance, do an insecticide application and to be sure that you are just suppressing them. Because from what I've seen in the field, I have more other insects rather than Philaenus spumarius or Neophilaenus campestris. So I think that now the point should be to try to be more effective on what we know. Like we have the models, we have the insecticides, we have other type of control methods, novel methods, but we really need to go further to have or to achieve better efficiency of those treatments, because we already know everything about the seasonality of the vectors, abundances, and the month that we can apply, not whatever.
Okay.
I wouldn't be so optimistic. I'm sorry, but I'm a pessimistic person. About the models, degree days and phenological stage, we have here a poster that is kind of different in the outcomes respect to yours, and basically we have this big black box that we are discussing with Gianni Giglioli, that is the modelist of the egg diapause, that we still don't know what are the climatic environmental determinants that stop it. And then, okay, the degree models, probably, okay, we can change some threshold, whatever, but then probably they work. But if we don't understand these, probably our models wouldn't be so effective at the large scale. This is my thinking. But, I mean, this is also a scientist that is talking with another scientist. Let's get practical. Maybe we don't need such a precise timing. Okay, you have to treat in this way to reach a good or enough control. And we are seeing that in Apulia the things are evolving in quite unexpected ways, or peculiar at least, almost resolving themselves alone. No, probably not, but I don't know. Probably the vectors are not the most important piece of the puzzle of Xylella. Is one of them. Yeah, we don't have. I'm an entomologist. I tend to think of insects before everything.
I wouldn't go there. No vector, no disease. So,
yeah, sure.
Yeah. Okay. Yes, it's true that one of the key elements in this model that I've been talking is when does the diapause stop? And this is important to know because depending when the diapause breaks, this accumulation of degrees could be different from one place to another. So to me, it's very important to know in each region when does it stop. And this can be also monitored. I mean, by obtaining field data, no? By obtaining enough field data. But yes, the determinants for diapause breakage are not known. And I think it would be very nice to understand why this diapause breaks.
All right, there's a question here, but it might be nice to figure out what's preventing you from just trying it out, these various models, and trying it today rather than tomorrow. We have a time for a very quick question over there and a very quick question over here. Clock's ticking. Oh, okay. Here first.
So Alberto, can we send you the data that we publish in EFSA, but we collect a lot of data from Italy, from Balearic, we collect from Portugal now about the biology of vectors. We can send you the data, you try the model, if it can work also for that area.
The good thing of this model is that you can predict it year by year. When is it going to happen? So yes, for validation would be nice to have that data.
All right, we may have a solution here. Last question.
Piotr Trembitsky from Macquarie University in Sydney, Australia. So when I was asking the previous question, sort of referring to the management as well a little bit, and I think maybe that's a bit too much to ask, because we do have like a model system with aphids and viruses that we cannot control, and they've been there in the environment for like decades. So, and I know there's lots of different models that can predict the timing and likelihood of establishing and stuff like that. But like in the real world, for the grower and stuff like that, I think some sort of threshold about how it actually economically is viable to treat or not. It's extremely important. So my question is, we have this information about likelihood of the Philenus to emerging in certain areas, but do we have information about the extent of it, and what does it mean for the spread of Xylella? Because I guess we can't just spray chemicals across board everywhere. We have to have some sort of more information about actually economically viable and it's going to make a difference. Do we have this type of information across? And I guess that's my question.
I think we don't have, but...
No.
Okay, so I think we all need coffee, but don't run away right now. We have a small movie on the plant health campaign of EFSA, right? Yeah, so 2-3 minutes. So thanks to all of you for your nice contribution and for the audience for the nice question, and enjoy the break. And we meet at 4:30.